Transcription is the process of DNA being made into an RNA copy. The RNA that's being copied is complementary to the template strand and similar to the coding strand, which is also called the non-template strand. After the RNA strand is transcribed, it is then turned into messenger RNA, or mRNA.
mRNA is made through the process of RNA splicing. The term RNA splicing is the process of introns of the pre-mRNA getting removed and the exons joined together for further translation into proteins. Exons are segments of RNA where the coding sequence of polypeptides are located. Introns are segments of RNA that need to be spliced out. There are two splicing mechanisms, constitutive and alternative splicing. Constitutive splicing is when one pre-mRNA splices to form the same mRNA each time and every time it is transcribed. No variation occurs in terms of exons being skipped. Alternative splicing is when the same pre-mRNA transcript yields multiple mRNAs. This process happens because multiple organs need multiple variations of the proteins. In an alternative splicing process, all the introns get removed, but not necessarily all exons get joined together. Some exons might be selected out of the protein depending on the need of the proteins. Notice that the order of exons never changes. How does this happen? Exons are joined together through the addition of a cap and a tail. A 5' cap is inserted into the beginning of the pre-mRNA. This cap consists of a modified guanine nucleotide that is added by the enzyme guanylyl-transferase into the free phosphate group. A poly A tail is added to the end of the pre-mRNA. This tail functions to protect the mRNA from getting degraded and aids to push the mRNA into the cytoplasm once splicing is complete.
From constitutive and alternative splicing mechanisms, three pathways arrive. Group 1 and Group 2, which are self-splicing, and one splicing pathway that happens by means of a spliceosome. The splicing of Group 1 and Group 2 introns occur via self-splicing, which is splicing that does not require an aid of catalysts. Instead, the RNA functions on its own ribozyme. Ribozymes are RNA molecules with catalyst activity. Group 1 and Group 2 differ in the way the introns are removed and the way exons are connected. The splicing of Group 1 intron involved the binding of a free guanosine to a site within the intron, shown here, leading to the cleavage of the RNA at the 3' end of exon 1. The bond between a different nucleotide in the intron strand and the 5' end of the exon 2 is also cleaved. The 3' end of exon 1 then forms a covalent bond with the 5' end of exon 2, which makes the RNA. The splicing of Group 2 introns are very similar to Group 1, except the 2' hydroxyl group on the adenine nucleotide begins the catalyst process, and it followed the same mechanism as Group 1 to make the RNA product. Pre-mRNA splicing requires the aid of a multi-component structure known as a spliceosome. Spliceosomes are composed of 5 subunits known as SNRMPs, pronounced SNRPs. During splicing, the subunits of a spliceosome carry out several functions. First, spliceosome subunits bind to the intron sequence and precisely recognize the intron-exon boundaries. In addition, the spliceosome must hold the pre-RNA in the correct configuration to ensure the splicing together of the exon. And finally, spliceosomes catalyze a chemical reaction that causes the intron to be removed and the introns to be covalently linked. This concludes the splicing mechanism in the termination phase of transcription with its 2 types, alternative and constitutive, and its 3 possible pathways into mRNA, self-splicing through Group 1 and Group 2, and splicing by spliceosome. The end.